my cutest little children. Good evening to everyone out there. I hope I am audible, I am visible. Yes, I am perfectly audible and perfectly visible. Just give me a second. There's a technical thing that I have to do. Papa. Yes. Okay. So we'll just wait for some time so that everyone else can also probably come and join our class. Okay, and then we will begin off with this particular session. I hope everyone is doing absolutely good. Okay, so my dear children, I hope we are ready. Listen to me very carefully. We have completed two parts of this particular chapter. Part 1 was where we spoke about the initial, introductory part. We spoke about the monohybrid cross and all those things, okay? And in this particular part itself, we spoke about the exceptions of the laws of inheritance. You know, after monohybrid cross was being done, very, very different experiments were being conducted. For example, the Snapdragon experiment of incomplete dominance and things like that. So all of it is being covered over here. Pleiotropy, polygeny. So if you have not watched that, make sure you go and check it out once. Okay, make sure you go and check it out once. Now the second thing that you have over here is going to be the part 2, where dihybrid cross linkage, recombination, sex determination, everything was being covered. Then I hope it makes sense and I hope it's clear to all of you. Now we're going to go ahead with today's session, where we're going to talk about the Mendelian disorders. This is one of the most important topics out there. This is the one that gives most number of questions as well. Okay, so we're going to categorize them into two parts. Okay, one is going to be Mendelian and one is going to be chromosomal disorders. What do you mean by that? The thing is very simple. Listen to me. Over here, suppose you have a chromosome. Okay, this is the chromosome that you have. On this chromosome, what do you have, Baba? You have genes. What do you mean by that? Basically, chromosome is nothing but your DNA, which is made to be very compact, right? And each part of your DNA might be coding for some protein. You call these parts as what? Genes. Suppose for some reason there are some issues with these genes out there. Suppose for some reason there are some issues with these genes. What is going to happen? Do they produce the same type of protein? They can either produce an abnormal form of protein, or probably they might not produce anything at all. Either way, that's a problem for us. Either way, that is going to be a problem for us, because all the proteins which are produced in our body are very important, right? These type of disorders which are being caused because of some mutations, some changes in the genes, are known as Mendelian disorders. They're going to be known as Mendelian disorders. On the other hand, just imagine, we need to have a pair of chromosome, one from each of our parent, one chromosome from one parent, another chromosome from another parent. This is how it generally works, right? So we get a pair of chromosome at the end. Suppose one extra chromosome thought that today I'll also go. In that case, you have an extra chromosome right over here, right? Got it? Over here, because of that extra chromosome, you might have some extra proteins being created. A lot of issues could be happening. This can lead to what? A chromosomal disorder, because of something that is extra over here, a whole chromosome that is extra over here. By the way, Harold, hi Harold. And next thing that can happen is probably, instead of having two chromosomes over here, we just have one is taken away by something. Okay, we just have one. Loss of chromosome can also lead to what? Chromosomal disorders. Or probably it can be something like this also. You have only one arm of the chromosome, the other arm is missing altogether. That can also lead to what? Chromosomal disorders. So if at all you see over here, everything is related to what? The issue regarding either the structure or the number of the chromosome. Over here in Mendelian disorder, the issue is regarding what? The alteration or mutation in a single gene which is there on the chromosome. Got it? It's as simple as that. Now we're going to talk about each of them in detail one by one. Before that, let me give a hello to all my dear children. Hello user SB7. Hi, I hope you're doing good. I hope you're doing great and you're full with energy. So my dear children, this is going to be the last part of Principles in Inheritance and Variation chapter, and I'm really excited. The reason being that if probably you're watching this, I can guarantee that you are going to score a good amount of marks from this particular topic. Okay, you have to be very careful, but that's the only thing. Got it? Works? So I hope this is clear to all of you. That is what is Mendelian disorder and what is chromosomal disorder. At least the difference is clear. If clear, can you give me a thumbs up such that I understand my children are getting things. If it is clear, can you quickly give me a thumbs up? User SP, ma'am, please explain on the topic Drosophila melanogaster. Dear, if at all you can, I'll be just asking you one thing, that is, if at all you can, there is a video that I did over here. Where is it? This part 2 video. In that I had explained about linkage and recombination, where I spoke about Drosophila melanogaster. It took me a lot of time to explain that. That's why I do not want to invest the same time over here. It will be waste of time for others. So if you can, please make sure you watch that, or probably we have to wait until the end. Okay, then I can probably revamp it once more. Will that work? And ma'am, when will the live end? Live will possibly end by either 6 o'clock or 6:30. That's the maximum time. Cool. Works? Now listen to me. Now, perfect. Sandhya, good evening, ma'am. Board has released multiple choice question. Actually, I have to see it. Once after I check it, I'll definitely let you know whether it's KCET level or not. Okay? I got to have a knowledge about it, I heard about it, but I have not checked it yet. So it will be very wrong if at all I give you wrong information, right? So I'll check and tell you, Sandhya. Will that work? Okay. Fine. Now listen to me. There is color blindness as the first Mendelian disorder. Now before we go ahead with Mendelian disorder and things like that, I want to put your attention to one more thing. Okay, because you might be seeing sex-linked disorders, you might be seeing some autosomal disorders, and you might get confused. So listen to me. In our body, how many chromosomes are there? Everyone, come on, guess. How many chromosomes are there in our body altogether? Full, complete set of chromosomes. You're most welcome, Sandhya. How many chromosomes are there, bacha? 45 are. Whereas the one chromosome out there, 46 chromosomes are there, okay? We have 46 chromosomes, and when we say they are individual chromosomes, they are 46. But when they form pair, 46 of them, when they form pair, 1, 2, 3, 4, like that, how many pair will you get? You will get almost 22 pair of chromosome. 23 pairs of chromosome. How many? 23 pairs of chromosome is something that you get. Now listen to me very carefully. Until 22nd pair, they have nothing to do with the sex of an organism. What do you call them? You will call this as autosome. Okay, you are going to call this as autosomes. They do not have anything to do with the sex of an organism. On the other hand, when you talk about the 23rd pair, the last one which is there, they are responsible for the sex of an organism, which is why you call them as what? Sex chromosomes. Sometimes you have another name for it. Can you guess what is the other name for the sex chromosome? Just like how we have autosomes, there is another name for sex chromosome. Anyone has any idea what do you call it as? Come on, guess. It also starts with A. It also starts with A, and you are free to Google it if at all you want. You are free to Google it if at all you want. I want you guys to seek for the answer. What do you call the sex chromosome? What is the other name for sex chromosome? That is the last pair of chromosome that is there. It can be XX in the case of a female, okay, or probably it can be X chromosome and a Y chromosome in the case of a male. So what do you call these chromosomes as? Come on, children. Waiting for the answer. Try, Madi. Otherwise, it is not going to be fun. If I only tell everything, it is going to be a boring session. You will get bored of me. No? You want me only to tell? Then fine, then I'll only tell. I'm going to tell, ready, 1, 2, 3. It's known as allosome. They are also known as allosomes. So we have autosome and we have a pair of allosome right over here. Now listen to me. Why did I speak about all of this right now? The reason being that if at all any of your autosomes gene is being affected, which is why you have a disease, you will call this as an autosomal disorder. On the other hand, okay, autosomal disorder. On the other hand, if it's your sex chromosome and a gene in the sex chromosome is affected, you will call this as sex-linked disorder. Got it? Autosome linked disorder will be on the genes on the normal chromosome, that is autosomes. Here, if at all a gene has an issue, okay, and that is in the sex chromosome, you will call this as sex linked disorder. So in Mendelian disorders, you might have seen something like this, that basically over here, you have so many diseases such as color blindness is there, hemophilia is there, phenylketonuria is there, so many diseases are there. and they have been tagged as sex-linked, autosome and things like that. And the first disease that we have is going to be color blindness. Color blindness is actually a very partial disease, Same thing goes with all the sex-linked disorders. I'll tell you the reason why. Okay, so listen to me carefully. Color blindness is a disease which is caused because of defects in the cone cells of your eye. The cone cells are responsible for seeing the colors. Okay, so there is red cone cells and there is basically green cone cells which are being affected specifically in a case like this. People can have different type of color blindness. I'm talking about the red-green color blindness. If your red and green cone cells are affected, basically you will be colorblind. You will not be able to see those colors. And this disease which is there, this affects more males than females. Yes, if I take a ratio, if I just take a percentage of things, you know what? Almost 8% of males are going to be affected. 8% of males, and on the other hand, when we talk about females, only 0.4% of females. It's not even 0.8% over here, it's 8%. 8% over here, it's 0.4% over here in the case of females who are going to be affected. The reason being what? The reason is that it is an X-linked disorder. Up until now, people are like, ma'am, I didn't get anything. Can you explain what do you mean? So listen, I get you, I totally get you, and the reason that you're not getting is because we do not know the logic behind it. So listen to me. When I say it's an X-linked disorder, that means your X chromosome has to be affected. In females, how many X chromosomes are there? Baba, in females, we have 2 X chromosomes. We have 2 X chromosomes. What about in the case of males? Males just have 1 X chromosome and the other one is going to be Y. Suppose, I give you a scenario. In the case of females, suppose one of the X chromosome is affected, and I'm just writing it like this. 1 X chromosome has a gene that is affected. What is going to happen? The other one is there to overshadow the effect, and this person is going to be having a normal thing. She's not going to be colorblind, she's just going to be carrier. Carrier means she has the gene, but she's not going to be colorblind. She's going to be having a normal vision. Okay. On the other hand, over here, if I talk about men, if one of his chromosome is being affected, X chromosome is affected, what is going to happen? Will he have normal vision or not? Will he have normal vision or not? Only 1 X chromosome is there. There is no one to overshadow the effect of this, which is why Papa guy is going to have what? Color blindness. Papa guy is going to have what? Color blindness. You get it? Okay. On the other hand, when you talk about females, again over here, females, suppose, how do you think, when will they become colorblind? They are going to become colorblind only when both of their chromosomes are affected. In that case, there is no one to overshadow the effect of the other, and which is exactly why this condition is going to be leading to what? Color blindness. Got it? That is why I say males are being affected. Now still, a lot of you might be thinking that, ma'am, I'm not getting it. Please give a scenario. So I'm going to talk about few different scenarios, and we're going to see who are the ones who are mostly being affected. Okay, we know the logic, we know everything, right? So we can probably go ahead and discuss more about it. So this is the first scenario that we have. First scenario. A person is there who is very normal, a normal guy, does not have any issues. Okay, then there is a female. Then there is a female who is going to be what? She is a carrier. Carrier means she has the gene that is affected. Okay, now both of them, if I have got a parent like this, I do not know my mother is colorblind. Why? Because she is showing normal vision. She does not have any problem. She is having normal vision only because the other chromosome is there. Okay, but she is a carrier. We know because we are making it right now. We know the genetics of it. Right? Now when you make the babies, let's make the babies. One gamete gets X, another gamete gets Y. One gamete gets X, another gamete gets XC, the affected one. When this and this comes together, you get XX. What is it? It's going to be a normal female over here. Very normal female. Done. Okay, suppose this and this comes together. Again, there is a female over here. What will be she? She will be a colorblind person? No. Why? Because the other chromosome is there to overshadow the effect. So can I say she is a carrier? She is going to be what, Baba? A carrier. Okay. Now the next scenario. This and this comes together, you get XY. What about this guy? This guy is going to be a normal male, ma'am. No effect, no problem. Normal male, okay. What about this and this? When it comes, XC comes and a Y comes. What happens to this guy? What happens to this guy? Come on, you tell me. Will he be colorblind or not? Will he be colorblind or not? One X chromosome is affected. Do you think he has anything to overshadow the effect? Do you think that they have anything to overshadow the effect? Will that person be colorblind or not? The answer is yes. This person is going to be what? Colorblind. Who is he? It's a male who is going to be colorblind. Out of 4 progenies that you see over here, who is the only one that is colorblind? The male is the colorblind. Now the question can be like this. Okay, a normal vision person marries another female who has a normal vision. Okay, what type of progenies will you get? So when you say normal vision, so you'll think that it is going to be XX, but always remember she can be like this also. She can be like this also. And if she is like this, Baba, you are going to get something like this. And in this 1, 2, 3, 4 progeny, how many of them are colorblind? Only one is colorblind. So 1 by 4 is equals to what? 25% of them. I can say 25% of total children are what? Colorblind. Now if you're talking about the boys, the son out there, in that particular case, what is going to happen? In that particular case, Baba, if there are 2 sons, this son, out of that, half of the sons are being affected. So if we just talk about the son, it is going to be what? 50% of the boys are being affected over here. What about the female? Female is either a carrier or she is not affected at all. This is just the first scenario. This is going to be the first scenario. Now here is a question. Okay, just a second. Ma'am, when is provisional and final result of KCET counseling engineering colleges? Okay. So when is provisional and final result of KCET counseling? They are yet to announce that. So tomorrow it's going to end. Most probably within this week itself you should be getting the provisional results as well as the final results. But cannot give you a guarantee about that. Okay. We are late for college. The college is going to definitely take into consideration about that. Dear, do not be worried. Probably they might be doing some arrangements such that you do not miss out on a lot of things. Okay? Yes. Done. I hope this is clear to you, children. This particular portion until now is clear. This was just first scenario. Now, if you guys do not talk to me, it's going to be really, really boring. Okay? It's not going to be fun. My energy will go down. You know what? I have been standing for so many hours and I am here in the live to talk to you guys. And if you are like, let the teacher teach, I'll just listen, it will be very boring. So you have to interact with me. I'm going to go ahead to the second scenario and this time you have to give me the answer. Okay? The second scenario works like this. The second scenario works like this. I have a person, a man who is having normal vision. Okay? And a female is there who is colorblind. Who is colorblind. What is going to happen? What about their kids? How many of them are going to be colorblind? Let's check it out. Okay, so over here X chromosome, here you have Y chromosome in the gamete, here you have XC in the gamete, here you have another XC in the gamete. Cool. When this and this fuses, what do you get? XXC. What is she going to be? Is she colorblind, Baba? Do you think that this particular lady over here is going to be colorblind? Understand that she has one chromosome which is normal. And then only you have to give the answer. Will she be colorblind or will she be a carrier? Come on, you guys tell me. This time I'm not going to tell you. Okay? This and this fuses together. You have again X and XC. Again a female is over here. Again, is she going to be colorblind or is she going to be just a carrier? Carrier. Sakiva gave me the answer. Thank you so much. A senior is coming and giving the answer. Juniors, come on. Okay, over here she is going to be what? A carrier. Similarly here also she is going to be a carrier. But let's check out the males. Papa guys, what is going to happen to them? Let's check it out. This and this comes together, XCY. Okay, this and this comes together, XCY. In both the cases, Baba, there is no X, another X2 probably overshadow the effect, which is why what happens to the males? If we check the males, what happens to the males? Both of them, are they going to be colorblind or they can be carriers? Do you think that they can be colorblind or do you think they will be just carriers? Such a sad life they are having. They are always going to be what? Colorblind in this particular scenario. Why? Because there is no other X to overshadow the effect, which is why, Baba, they are going to be colorblind. Now in this scenario also, ayyo, what is happening? Just give me a second. Yeah. Ayyo. In this scenario also you see males are affected more. How many males? Actually, if we talk about the total organisms, okay, the total progeny which are there, out of 4 children out there, 2 of them, okay, 2 of them are getting the disease. 2 of them are being affected. So I can say 50% in total of them have the disease. Now when you talk about the boys, if you're talking about just the sons, how many sons have the disease? If that is the question, then there are only 2 sons, both of them have the disease. So I'll say 100% of the boys will generally have the disease. Here also boys are being affected more. Yes, Sakiba, they are being affected over here. Right? In this both scenario, we had made the boy to be normal, the father to be normal, and the mother was a carrier, or basically she was colorblind. Now let's twist the story a bit and probably go ahead with making the father colorblind. Let's see what is going to happen in that particular case. Okay, I'm going to make the father colorblind and the mother we are going to make normal. Let's see what is going to happen. So father is going to be colorblind over here, and the mother who is here, she is normal. What is going to happen? Listen to me very carefully. Father cannot give this particular gene to his son, can he? Because father, when he makes a son, he only contributes the Y chromosome. So sons are not going to be affected because of him. I'm just giving you a heads up. Let's check it out once. Okay, so over here I am going to make the chromosomes. X in one, Y in another, X in one, and another one also X. This and this comes together, what do you get? You get XXC. This is going to be what? A normal female, a carrier one actually. A carrier, why? Because this one overshadows the effect, but she is still a carrier. She has a defective gene. So she is going to be a carrier female. Okay, what about this and this? Again, she is also going to be a carrier female. What about this and this? XY, normal men. XY, normal men. If at all you see, none of them have the disease, even though the father has it. Even though the father has it. Why? Because over here, only the father has the gene, and the father cannot transmit it to the sons. Even if he transmits it to the daughters out there, they are going to be normal. Why? Because the mother does not have it. Okay, let's think about one more scenario. It was pretty easy. Okay, let's think about one more scenario. Hi, Amalu. It's been a long time. How are you? I believe, a person with a similar name was there previously also. So I'm good. How are you? Okay, anyways. So over here we have XCY. Here, the man who is there, the father who is there. Baba, he is colorblind. Okay, what about the female? Female is going to be a carrier. Let's check what is going to happen in this particular scenario. This is going to be XC in one particular gamete. Okay, here it is going to be Y in another gamete. Here it is going to be X in one gamete. Here it is going to be XC in another gamete. Cool. Now when this and this comes together, you get XXC. When this happens, is it going to be a carrier female? Yes. Why carrier female? Baba, if at all you see, she has a defective gene, but she is not going to have color blindness. Why? Because there is a normal gene to overshadow the effect. So I'll say she is a carrier female. Okay, no problem. What about this and this? When both of them comes together, and both of them is affected gene, will that person, okay, will that organism be, what do you say, 50% affected and 50% normal? Sakiba, percentage. Is it? Okay, we're going to check that. Amalu says, I'm fine, ma'am. Long time no see. Good to see you. Good to see you, dear. Actually, I'm also really happy to see you over here. Okay, so let's check whether Sakiba, whatever she is saying, is correct or not. So over here, the female who is there, she is having the disease. So I'll say she is going to be colorblind. She is going to be color blind female. Okay, let's check about the males. This and this comes together, XY. It's going to be a normal male. This and this comes together. This person is going to have the disease. It's a male, but who has the disease? Why? Because there is no X chromosome to overshadow the effect. So again, you get colorblind. Okay, this is going to be a colorblind male. So as Sakiba said, 50% of them are going to be affected. If you take 4 of them, 2 of them are affected. So 50% are going to be affected over here, 50% are going to be normal. Right. In this particular case also, if you see the males in general, if at all we spoke about all the scenarios, we have noticed one thing: the males are the ones which are usually affected. Why? The reason being that one defective chromosome is more than enough for a person to have the disease. Now we have one last scenario, which I don't think you need to make it, but still, if at all you feel it's necessary, let's go ahead. The person over here is a colorblind man, and the female over here is a colorblind female. Let's see what is going to happen in this particular scenario. This is going to be the last scenario which is out there, and definitely this is going to be helpful also for us to understand a lot of things. Okay, XC in one, Y in another. XC in one and XC in another. What happens? Do we even need to cross it? Do we even need to cross to check who are going to be colorblind or not? So when we cross this and this, you get XC, XC. Here this person is going to be colorblind. Here this person is going to be colorblind. Why? Because both the chromosomes are affected, and this is going to be a colorblind female. A colorblind female, right? When you cross this and this, again, when you cross this and this, again it will be XC, XC. It is going to be colorblind female again. When you cross this and this, it is going to be XC, Y, which is going to be what? A colorblind male. When you cross this and this, it is going to be XCY, which is going to be a colorblind male. All of them are going to be what? Colorblind. Amalu says all are colorblind. Exactly, all are going to be colorblind. Hello Kumar, I hope you're doing great. Okay, so that is a logic. Now if you see this scenario, here the male is affected, here all the males are affected, okay, and here in this previous scenarios that we studied about, okay, in this particular case also, males were the one who were generally affected. We know the reason for that because it's an X-linked disorder, and in X-linked disorder, even one defective allele which is there, if it's there and you do not have anyone to overshadow the effect, it will be showing up the disease, right? Right. That's the only thing that we need to know. Guys, just give me a second, a quick second. Okay, now I believe I'm audible, right? Okay, so Kumar, what's your name? My name is Jasmine, and Dost, hello dear. Hello, hello, hello. Okay, so we were talking about color blindness, and we studied that it occurs in only 8% of males. It occurs in 8% of males and only 0.4% of females. I hope you know why now. So females are usually carriers unless they inherit two defective copies. For them to inherit two defective copies, one from the father, one from the mother, basically the father has to be colorblind. If at all the daughter should be colorblind, the father has to be colorblind. The mother can be a carrier, or she can also be colorblind. Okay, either ways you can get one particular thing, but you have to get one from either of the parents. Now probability, a carrier mother has a 50% chance of passing it to a son. A carrier mother, over here we saw the scenario, this was the carrier mother, she has a 50% chance of passing the gene to her son, right? To her son. Why? Because the mother is the one that is going to contribute the X chromosome, father contributes only Y. So 50% of our X goes like this, normal, 50% of defective X goes to the other son which is there. So 50% chances there for a son to have the disease, right? Right. Now finally over here, what do we have? A daughter will be colorblind only if her father is affected and the mother is at least a carrier. If mother is at least a carrier, then probably it works. Okay, understood. June says understood. Thank you. Okay, ma'am, what will be there? The next generation happen for all colorblind, right? Which one, dear? Which one all colorblind? If you have both the parents to be colorblind, then only you are going to see color blindness in the next thing. So if you see that both the parents are colorblind, in this all the progenies are going to be colorblind. Cool? Dost, if I was a bio student, then I would have got a chance to study from Jessie ma'am. I am Jasmine ma'am, and and I'm so glad you said that. Thank you so much, dear. That's actually sweet. Okay, so I hope it makes sense, right? Guys, do you have any confusions around color blindness, or can we move around with the next topic, that is hemophilia? Any doubt, June? Yeah, next generation for that 4, basically, if suppose, okay, next generation for that 4, as in, for example, I think, yeah, over here, suppose I talk about this one, I talk about she. Suppose she marries a normal man, then what happens? If she marries a normal man, if she marries a normal one, what is going to happen? Let's see the scenario, okay? I've already made it. If she marries a normal man, 50% of her son, sorry, all the sons over here are going to be affected. What about the daughters? Daughters are just going to be carriers. Daughters are just going to be carriers. So it is going to differ, okay? It is going to differ upon the, what do you say, partner, depending on the partner. If he is colorblind, things are going to be different. If he is not, things are going to be different. Got it? So this was a scenario where he was not. When he is not, basically, as you can see over here, daughters are not going to be affected, but the sons are going to be affected. Works, June? Understood? Yes, ma'am. Yeah, most welcome. Okay, so that is about color blindness, and now we are going to go ahead with a question. A KCET question. This is actually a very recent question, KCET 2025 question. A colorblind man marries a carrier woman. The percentage of their colorblind progeny in the next generation will be what? That is the question. So over here, let's decode it one by one. Colorblind man. Colorblind man means it is always going to be like this. This person cannot be colorblind because these are normal. Okay, colorblind man looks like this. Okay, here he marries a carrier woman. That is also mentioned. Carrier woman means one chromosome has to be affected. Then she becomes a carrier. Now the question is what? Okay, Kumar says the answer for this question is 25. Let's check it out, Kumar. Okay, let's check it out. Your question is, what will be the percentage of colorblind progeny? Okay, so this chromosome goes to this gamete, this chromosome goes to this gamete, this chromosome goes in this chromosome goes in this. When I fuse this and this, I get XXC. What happens over here? She is going to be a carrier, not colorblind. She is going to be a carrier. Suppose this and this goes. Suppose this and this goes. What is going to happen? Over here, both the chromosomes are affected. Both the chromosomes are affected, which is why what is going to happen? She is going to have the disease, right? She is going to be having the disease. I'll write that she is going to be colorblind, and this is a female who is going to be colorblind. And I make a star mark so that I do not forget. Now this and this comes together, XY. This is going to be a normal man. None of the genes are being affected. And finally over here, I get a man. Okay, I get a man who has one X chromosome being affected, but for a man, one X chromosome is still fine to be affected. This man is going to be what? Color blind. And this is a man. The question is, how much progeny, how many percentage of their progeny is going to be colorblind? So if they have 4 children, out of 4, 2 of them are affected. 2 of them are affected. 2 out of 4 means 1 by 2. 1 by 2 means what? 50%. In that case, what will be your answer? Amulu says the answer is B. I saw a lot of you remove the answer. The answer for this question is going to be what? B na macha. But it's okay. If you got it wrong, it's still fine. Why? I'll tell you. That's how we learn. Okay, that's how we learn. So Kumar now said B. Perfect, Macha. The answer is going to be what? 50%. Sometimes you have to be really careful. They will ask you how much percentage of the females are affected. If the question is, how much percentage of females affected, what will be your answer? So females are 2. Out of 2 females, if one is being affected, you say 50%. Okay, out of 2, 1 after 2, basically 50% of female is affected. Over here, 50% of male is also going to be affected. As simple as that. Okay, works. This question is clear to all of you. Only if it is clear, we will move on to the next question. Only if it is clear, we will move on to the next question. Clear? Give me a thumbs up. Okay, okay. June says okay, Kumar says okay. Okay, fine. Let's go ahead then with the next question. This is a 2018 question. It goes like this. Both male and female have normal vision. Both male and female have normal vision. Now I always tell you, okay, always tell you one particular thing, that is, for a male to have normal vision, he definitely has to be like this. He cannot be a carrier anyway, so he'll be like this. What about a female? A female, when I say normal vision, in the back of your head, always know that she can be like this, or she can most probably be like this also. This person will be a carrier, but she will have a normal vision, right? Right. So have that in your head, okay? So I have written about the male, I have not written about the female. I'm confused. How do I clear my confusion? By talking about her parents. Let's talk about it. So both male and female have normal vision, though their fathers were colorblind and mothers did not have any gene for colorblindness. Listen to me carefully. If the father is colorblind, father always contributes his X chromosome to the daughters. Okay, and mother is not colorblind, that means she is like this. If this father is contributing X chromosome, From the mother only you get X chromosome. She is definitely going to be a carrier. There is no other option. Okay, if the father is colorblind, she is going to be carrier. So people are giving me the answer as A. June says A, Amalu also says A. So probably I have to check it out. Okay, wait. So this person is going to be a carrier. I hope this is clear why she is a carrier, because that is written her father was colorblind. That means she has to be carrier. Mother is not having any gene for colorblindness. The question is the probability of their daughter becoming colorblind. Okay, X in one, Y in another, X in one, XC in another. When this and this crosses, you get XX. This is a normal female. Okay, when this and this crosses, you get XC. This is going to be a carrier female. The question is the probability of their daughter becoming colorblind. None of them are going to be colorblind, which is why the answer is going to be what? 0%. It is going to be what? 0%. Your question was the probability of daughter. You were not being asked about the sons and all. If you're asked about the sons and basically whole of the progeny, it will be XY, a normal man. Here, XC, the only man who is having disease. The total percentage, if you are being asked, it is 25. Out of 4, only 1 is affected. That's when we say 1 by 4 means you are talking about 25%. But that was not the question. The question was just about the daughters becoming colorblind. Daughters are not colorblind, so 0%. Kumar Baba, got it? Got it? Sajun, Amlu, everyone, thank you so much for participating. Kumar, thank you so much for participating. I hope you guys are understanding. If you're getting your answers wrong, it's because it's a new thing and that's very normal. Okay, don't be disheartened. You're learning and probably you will have that in your head. Okay, now if this question is super clear, just give me a thumbs up. I'll understand. We will move ahead. Okay, perfect. Thank you so much. Let's go ahead with the next question. Next question is not there. Next concept is there. So we've got 2 questions from this particular concept, that is basically color blindness. Now we move on to the next question. That is what? That is going to be next particular disease which is there, that is hemophilia. Before I go ahead with hemophilia, one very important thing. I request every one of you to definitely make sure you check out the all-in-one page. What do you mean by all-in-one page? So there is a page that we created, a website, a free website. You do not have to pay any money and all. There academically we are going to see, you will be able to see in bio what all things are being completed, in chemistry, in physics, in mathematics what is being completed, and basically the PDF notes are also being uploaded there. So if you wish, you can go check it out. In the chat session over here, this particular link was given. Apart from it, under every video, okay, so suppose over here I have one of my video, okay, the one short video on the chapter human reproduction. Below that, if at all I see over here, there will be a thing. Just a second. Join the free ACE KCET series right now. The free one which is there, you just have to click on that. Over here, click on buy now. It will be 0 rupees. Just put in your mobile number and all. You would be getting enrolled into it, so that you don't have to come to YouTube to probably check it out. That is known as all in one page. If you wish, you can check it out that. And if you do not want to miss out on any updates, make sure you join our WhatsApp channel as well. Okay, now let's come back. So we're talking about the next disease, that is hemophilia, which is again an X-linked disorder, which means that a person, a man who is going to be like this will have the disease. What about a female who has just one chromosome being affected? She's a carrier. She is not going to have the disease. For her to have the disease, you need both of her chromosome to be affected. So color blindness was actually a fatal disease. Color blindness which was there was actually not a fatal disease. You will not die because of color blindness. You are just going to live. You are not going to see those colors. That's the thing. But hemophilia is not like that. It's a fatal, it's a dangerous disease. Why so? Because what exactly happens in hemophilia is that, suppose there are so many, okay, just suppose there are so many, what do you say, proteins which are involved in clotting of blood. What do you mean by clotting of blood? Suppose over here I get a cut. Okay, I don't make a cut, I get a cut. When I get a cut, what is going to happen? The blood is going to go. Now I want the blood to stop. Our body to make sure that we do not lose out on blood, basically have some clotting factors which make sure that the blood does not go away. Okay, go away. That's the logic. So for that clotting, you need some factors, you need some proteins out there. You call these as clotting factors. Now, there are a group of proteins, among them one protein. One protein is being made by one particular gene on a chromosome, which is a sex chromosome. Suppose that particular thing is being affected. If that is being affected, what is going to happen? Clotting happens? No, clotting does not happen. So a person who has hemophilia, if at all they get a small cut, they're gonna bleed, and it can even lead to death. So they have to be very careful. Now your NCERT is pretty much old, okay? It's like your dada's age, that old it is, okay? So that's why they have not mentioned about the, the treatment offers that are there right now. But okay, right now people with hemophilia, they can survive. They can easily survive. But I'm just telling you, it's a fatal disease if at all it is not taken care of, if people are not aware about. Now, in your NCERT, there is a line, and that line says that the possibility of a female becoming hemophilic is extremely rare, because the mother of such a female has to be at least a carrier and the father should be hemophilic. What? Why is that? Why can't a female be hemophilic in nature? So they have mentioned that for a female to be hemophilic, one affected chromosome should be coming from the father. Another affected chromosome should be at least coming from the mother, so mother can be carrier, suppose. In that particular case only, this and this comes together and it will be like this. But the problem is that why this is very, very rare is because the boys, okay, the guys who are hemophilic, we know X chromosome linked disease, so boys are the one who are majorly being affected. So if boy is being affected over here, they might not survive until reproducible age. Okay, they might not survive until what? Reproducible age, such that basically over here they can reproduce and give rise to a progeny who is a female. That is the thing. That is why we say that this thing is very, very rare. But right now, it's not like that. NCERT has to change. It has not. Okay. Sham Kumar, good evening. Ma'am, is there any technique to stop the blood or not? So right now, yes. Okay. Does the clotting factors not work? So in our body, in people who have hemophilia, in their body, it does not work usually. But right now, according to the technologies that we have, yes, we have sources which helps in, inducing. You know, there are proteins which are being put inside our body, which is going to do the same function of clotting and all, which is why we could survive if at all we have hemophilia. But it was not the case before. Okay, it was not the case before. Okay, so that is why you consider this, it's still a fatal disease for people who do not know about the disease, and if they are having it, they are not aware about it, they are not getting the treatment and things like that. Okay, so I hope this line is super clear to you. If a female, for a female to have hemophilia, it's very, very rare. Why? Because the father has to have hemophilia and the mother should be at least a carrier. Okay, but the father cannot stay until the reproducible age to probably reproduce a female child out there. That was the logic before. Now it is changed, but at least we have to follow NCERT, so please do follow your NCERT. Clear about hemophilia. This is the second disorder that we have in sex-linked disorders. What are sex-linked disorders? Here the sex genes on the sex chromosomes are being affected. That is why you call them sex-linked disorders. Clear. Okay. Oh my God, something Hindi is there. मत कर मोहब्बत तेरे बस की बात नहीं, जो दिल को मोहब्बत वो दिल तेरे पास नहीं, अगर मैं होता बादशाह तो कानून ऐसा बना देता, दिल तोड़ने वालों को सजा ए मौत को माय गॉड सुना देता. Okay, so the कानून is not in your hand, so we can be happy right now because no one will get any sazas. But anyway, that's so sweet and that's so creative. Okay, now listen to me very, very carefully. Ma'am, does menstrual cycle bleeding also come to them? Menstrual cycle bleeding. So listen, that's what I said. For a female, okay, for a female, if she is hemophilic, she also will not survive. It's not just related to male, it's also related to females. They will also not be able to survive. If she is a carrier, yes, she will have a normal menstrual cycle because she is just a carrier, she has a normal gene. If she is a hemophilic person, yes, even menstruation could be a big problem. Okay, bol diya. I have told that shire. Okay, let's come back. Let's solve this particular question. The gene for hemophilia is located on X chromosome. Hence, it is normally impossible for a what? Hence, it is normally impossible for a what? It's asked about normally impossible. Okay, so gene for hemophilia is located on X chromosome, hence it is normally impossible. Amalu says the answer for this question is D. Okay, what about others? Come on, try. So, is it normally impossible for a hemophilic father to pass the gene to his daughter? If he is hemophilic, Baba, he can pass. It's just that he would not survive until that particular point. He definitely can pass. That's not an issue. Okay, so this is wrong. This is not impossible. Carrier mother to pass the gene to her daughter. If she is a carrier mother, okay, she can pass her gene to the daughter because yes, mothers can pass X chromosome. So this is also possible. Carrier mother to pass the gene to her son. Carrier mother can pass the gene to her son also because sons get their X chromosome from the mother. So yes, there is a chance that can happen. Okay, so June is saying D. Okay, viral genus is also there. Hello says the answer is D. Okay, everyone says D. Lastly, hemophilic father to pass the gene to his son. Hemophilic father, can he pass the gene to his son? No. The reason being that X chromosome, he cannot. Fathers cannot pass X chromosome to their son. They can only pass Y chromosome. That is the reason, which is why the answer for this question is going to be D. And my dear children, ta-da! Big salute to all of you because you guys are doing absolutely good. So super proud of you. Perfect. So happy to see this. And by the way, I hope everyone who is watching, if you're understanding, you have hit on the subscribe button as well. Perfect. Cool. So let's go ahead. Now we have a whole new disease altogether, and that is sickle cell anemia. Are we ready for it? Are we ready for it? So we just have two, sex-linked disorders. We covered that. It was color blindness as well as, hemophilia. Right now we have autosomal disorders starting at this point, and that is what? Sickle cell anemia. So if you're ready, just give me a thumbs up. We'll go ahead and we'll talk about that. Yes, ma'am. Okay, thank you so much. Let's talk about sickle cell anemia. So listen to me. Before I go ahead to talk about it, we need to have a briefing about things, okay? So what do we have in our body for transporting oxygen and all? What is that thing that we have in our body for transporting oxygen? Any guesses? There is one thing, a pigment, which is super important for transport of oxygen. Blood. Okay, blood is there. Okay, fine. Blood is there. In the blood, what do we have? In the blood, we have a cell which you call as RBC. Red blood cell. Okay, we have a cell which is known as RBC. June says hemoglobin. Exactly. Hemoglobin is something that I was looking forward for, where I'll also say blood. Okay, perfect. You guys are fine. Okay, we have in the blood, we have so many different type of cells. RBC is there, white blood cells are there, platelets are there, so many different type of cells are there. RBC over here is super important. Why? RBC is super important. The reason being that they have a protein within them which you call as hemoglobin. Hemoglobin. And this hemoglobin is the one that catches the oxygen and says that, Macha, you cannot go anywhere. I am going to transport you to different cells who needs you. You know, we all need oxygen. All the cells of our body, they are crying for oxygen. Why? Because only when oxygen comes, that's when the glucose can easily break down and we can produce ATP. And that ATP is our energy source for us. That is what runs all the things in our body. So if at all we are not getting oxygen, we die for the same reason, because in our body there will be no oxygen. Right. So hemoglobin is a very important protein that we have for basically what? For transporting oxygen. Transporting oxygen. Now listen to me carefully. This hemoglobin which is there is made up of two things. Alpha globin chain and another thing that is known as beta globin chain. Okay, alpha globin chain and beta globin chain, they are going to come together, and that's when they are going to basically go ahead and form the hemoglobin molecule. I hope this is super clear to you. Adenosine triphosphate, right ma'am? ATP, yes June. It is ATP, is adenosine triphosphate. My dear children, until this particular point, if it is clear, just give me a thumbs up. RBC has hemoglobin. Hemoglobin is therefore basically transporting oxygen. Hemoglobin is made up of alpha globin and beta globin. If it's clear, just give me a thumbs up. Then I'll go ahead. In our when, okay. Hansika, will it be fine if at all I read your comment a little late? If it's possible, and by the way, I'm making a video today. At 6:30, the video will be out for the same reason. Okay, perfect. Thank you guys for giving me a thumbs up. Okay, and Hansika, please don't mind. The reason is because there is teaching class ongoing, but I really want to give you the answer. I hope you understand. Yes. So my dear children, what exactly happens is that, listen to me carefully. Your body, okay, is going to produce proteins only if the genes on the chromosome gives the information to to form that particular protein. So over here, the protein that we are looking forward for is what? The beta globin chain. That is what is going to be made, right? And for that, this is a gene that has to be there. Suppose this gene gets some mutations, some changes happens in this particular gene. Do you think the same beta globin is going to be produced? There are chances there is no beta globin that is going to be produced, or basically it will be produced but in a very less amount. So what is the scenario that is going to happen over here? We're going to talk about that. Over here, the thing that is going to happen is beta globin is going to be produced, but it is going to be a mutated form of it. It is going to be what? A mutated, a changed form of it. What change, ma'am? I didn't get it. I'll tell you. So basically what exactly happens is, suppose this is our DNA. This DNA strand which is there, it gives information to make protein. But it cannot directly make protein. For that, first of all, it has to make something that is known as RNA. Your DNA transfers all the information to an RNA. This RNA is the one that gives a signal that, hey, this particular protein has to be made. And the RNA which is there, basically, it has some codes, okay? Just like how your mobile phone has chips and chips have all the information, similarly over here also you have certain codes. The codes get transferred over here to form the RNA. Here you have a code, suppose it is GAG. There's a code that is GAG. These codes which are there, they come in triplet order, okay? They come in triplet order. These triplet order, you call them as codons, and basically they code for amino acid. They code for what? Amino acid. Over here, the amino acid that this codes for is known as glutamic acid. Glutamic acid, what is this? This is nothing but an amino acid. Ma'am, why are we making amino acid over here? Listen to me carefully. How is protein being made? Protein is being made when all the amino acids comes together. That's when I say that I have got a protein. So over here, you need to use your imagination. Okay, you need to use your imagination. I am saying you have one codon coding for one amino acid. Similarly, don't you think you will be having multiple codons here and here on the either side? Yes, there are going to be multiple codons on either of these sides. They're going to produce what? Multiple amino acids as well. I am talking about one specific codon over here. This codon, this is the 6th codon out there. This is the 6th codon out there, which generally produces glutamic acid. Now, there is some change happening over here in the DNA, which is why you're gonna see that basically in this portion of the RNA there is going to be a change. Instead of GAG, here the codon has changed to GUG. You see, there's a change that was happening up over here, which is why now in the RNA also that change has been reflected. It is coding for GUG. What's the problem with GUG? What's the problem with GUG? It is also a codon, but it's not going to code for the same amino acid over here. It is going to code for another amino acid which is going to be known as what? Valine. It is going to be known as valine. Now what's the problem with that? The problem is that until now these amino acids were coming together. Over here right now a new amino acid has come. If a new amino acid has come, don't you think, okay, if that same protein is being produced, it's being produced, that is fine, but this time there is going to be a mutated beta globin that will be there. A different form of beta globin which is going to be there. If that's the thing, what happens? If that's a mutated beta globin that is going to kachara, bad one, that is going to take participation in the formation of hemoglobin, do you think the hemoglobin is going to be formed fine, or will it have some issues? What do you think? Will it have some issues or will it be fine? Hansika, please don't be sorry. Okay, I totally get you, and I'm pretty sure I might have given your answer in the video that I've already made, so make sure you watch it, and I'll try my best to tell the answer towards the end. I have taken a screenshot of your problem. Okay? Yes. So June says there will be issues, ma'am. Definitely, there is definitely going to be issues. Now, what issues? That is the next problem. Okay, what issues? So this hemoglobin, this bad form of hemoglobin is going to be where in your RBC right now? Right, this bad form of hemoglobin is there going to be in your RBC. This hemoglobin which is bad in nature, Baba, they are going to clump up. Now there is not just one hemoglobin, there can be multiple hemoglobin molecules out there. They all clump up. We say they polymerize, that means they clump up together, which is why their oxygen carrying capacity is going to reduce. Now they are not normal hemoglobin. Normal hemoglobin was doing the oxygen transport very, very nicely. Right now they cannot. They are not going to have They're not going to carry enough amount of oxygen, which is why the oxygen amount is going to be less. In this particular scenario, your RBC had never faced a situation like this where there was less oxygen. Now when it is facing a situation like this, what is going to happen? Your RBC's shape changes to something like this. That is, it becomes sickle shape. It becomes what? Sickle shaped. Now what's the problem in being sickle shaped? First of all, the reason why it was like this was to make sure that it has increased surface area for exchange of gases and things like that. Okay, so that is anyway not going to happen. Second thing, if it makes a structure like this, basically it can cause blockage, because structures like this can easily come together and create blockage throughout our body. So first issue was our body is not getting oxygen. Now it is creating blockage also. This is something that you see in people who have sickle cell anemia. Okay, so June is saying RBC shape will be changed, ma'am. Yes, enough O2 will not go to the blood. Exactly, enough O2 will not go to the blood. There will be a change in the RBC shape. Lot of problems are going to happen. And Viral, yes dear, there will be definitely issue. You get it? So this is the thing that is going to happen if a person has sickle cell anemia. Now, in sickle cell anemia also there are people who are carriers. Okay, and there are people who are not carriers. We're going to talk about that, but before I go ahead with any of it, since it's very, very new to you, I'm just going to revise it once more. Listen to me very carefully. Okay, just listen to me carefully. Over here, you have your RBC. Inside your RBC, you had hemoglobin. Hemoglobin actually has iron, which is responsible for trapping the oxygen and for transporting it. Okay, so hemoglobin, which is there, it is made up of two things, one alpha globin chain and one beta globin chain. Normally, that is how it works, right? Now, the beta globin gene which is there on one chromosome and autosome out there. Suppose this gene has been mutated, what is going to happen? Initially, the RNA that was there, it had a codon at the 6th position, that was GAG. Here there is a mutation that is taking place, which is why instead of A over here, you have got U. So GAG was a codon that was coding for glutamic acid, but now GUG comes into picture, it codes for valine. One amino acid changes in this at the 6th position and the whole protein has some issue. Okay, your whole beta globin protein has some issue. This beta globin bad one combines with alpha globin to form hemoglobin. Now the hemoglobin is also not fine. Hemoglobin is also not fine because the quality itself is bad. Right? Now the hemoglobin which is not fine will be coming together. All the hemoglobin molecules are going to come together. They are going to clump up, which is why they will not be trapping enough amount of oxygen. If they are not trapping enough amount of oxygen, what is going to happen? The level of oxygen decreases within the cell itself, which is why the cell becomes sickle shape. If the cell is becoming sickle shape, what is going to happen? They can clump up together and they can cause blockage. Got it? Now there's a way for you to remember this, GAG to GUG. Always know, in alphabetical order, A comes before U. So it was A first and it is replaced by U. Okay, in alphabetical order, G comes before V. So it was glutamic acid first, then it gets replaced by V, that is valine. And it is happening in the 6th position, do remember it. And this is the example of point mutation. This is the best example of what? A point mutation. What do you mean by point mutation? Point mutation means at a single base pair the change is happening. Only at a single point the change is happening, and that is this. Cool. And this disease is going to be what? This disease is going to be a qualitative disease. Qualitative disorder. What do you mean by qualitative? Basically the quality of it is being hampered over here. Okay, Rishi, definitely I do remember you. Oh my god. Ma'am, will agglutination occur, ma'am? Yes, agglutination means basically they clump up. Yeah, that can occur. How are you? I'm so glad to see you over here. I hope you're doing really, really good, Rishi. So happy. You could see that by my face, I think. Perfect. Okay, so that is a thing that is happening over here. Now I want to give you one more very important information. That is this chromosome that we were talking about. You have to remember the name of it, the number of it. This is chromosome number 11. Okay, this chromosome where your beta globin gene is there, you call this as chromosome number 11. Okay. Now listen, one more piece of information. How it is being transmitted, because as of now you just know that this is how the disease is being caused. How it is being transmitted. So here also, do you have carriers, ma'am? What is the scenario that we have over here? So to make you understand that, I need a page which I do not see, so probably I have to go like this. So listen to me carefully. Okay, here people can be carriers if at all they have both the alleles like this. We say they are normal human beings, they have normal hemoglobin. If we say that basically one is normal, the other one has some defect, we will say that this person is going to be a carrier. If I say that both of the hemoglobin has hemoglobin gene which is there, you know, there's a pair of chromosome. This is pair of chromosome number 11. Here you have a gene, here you have a gene for beta globin. If I say basically both of them are defected, I'll say that this person has the disease that is sickle cell anemia. Sickle cell anemia. Now I have a question. Two people, yes, Amalu, alpha globin is in the 16th chromosome. Yes, we're going to come up to that. Okay, if two people are having sickle cell anemia, will their progeny have sickle cell anemia? Obviously, yes. But listen to me carefully. Two people who have sickle cell anemia, will they survive until the point that they can reproduce and produce progeny? Most probably, no. Most probably, no. So usually we consider the cases of carriers. We consider that if two carriers are there, okay, two carriers are there, how much percentage of their progeny will have the disease? That is a usual question that is going to come. And if that question I give to you, what will be your answer? If basically I'm asking you this question, if there are two carriers, carriers means it is going to be like this. Carrier means they are going to be like this. If I cross two carriers, how much percentage of the progeny will be affected? Will be affected. This is chromosome number 11. I am telling you, one is a normal one and one which is there, it is going to be a mutated allele. They are the carriers that we have, and I've been asking you what happens. How much percentage of the progeny is going to be affected? Any guesses? Any guesses, or do you need explanation? I'll definitely explain, that's for sure. But can you guess also? Can you guess as well? One is to 2 is to 1. Okay, just a second. No, I'm not talking about the ratio. Okay, let's go ahead. Suppose this is going to be there. This is an allele, so it will be there in one particular gamete. This is another allele. This will be there in another gamete. Okay, this is one allele which will be there in one gamete, and this is another allele which is there in another gamete. Okay, suppose this and this comes together, you get HBA, HBA. This person is going to be normal. We do not know whether it's a male or female. Why we do not know? Because we're talking about autosomes. Whatever X chromosome that is being taken up, that is, if at all it's X chromosome that comes together in a person, or XY, that decides the, what do you say, whether it's a female or a male. So now June says 25, Ral says 25, and Amalu says 50%. Let's check it out, okay? So here it is normal. Here this and this comes together, HBA, and we have HBS. In this particular case, this person is going to be a carrier, right? Because one affected gene is there, one normal is there. This and this comes together, it is going to be HBA and HBS. In this particular case, one is going to be what? A carrier. Why? Again, one defective gene, one normal gene is there. Lastly, this and this comes together, you have HBS and HBS. If both of them are HBS, this person is going to have the disease. This person will be having the disease. They are going to be sickle cell, and they will be having the sickle cell anemia. So out of 4 of them, one is being affected. If one is being affected out of 4, so what do we say? 25% of the progeny is going to be affected. Okay, Amalu, super clear? Okay, June, correct. Okay, June, correct. And Viral was correct. Probably he just went. He was worried that it could be wrong. Okay. Clear, children? Clear, right? And never, I'm telling you, Evan, never ever be worried if at all your answer is wrong. Listen, no one who is probably watching this video with you have seen your face or probably knows you personally, and if at all they do also, it's not an issue if you give a wrong answer. Why? Because what I believe is people who try, they are better than people who do not try and just judge, right? So if you try and get it wrong, it's okay, you get it better. You remember that I did a mistake. You probably remember things better. You're explaining very clearly, ma'am. Thank you. So sweet of you. Thank you, June. We're all listening 25. Okay, got it. So this is a scenario. Now I'll tell you one more very important thing. This is a reason why people say that we should be, you know, people when they get into marriageable age group, okay, when they get into marriageable age group and when they plan to have babies, they just have babies. They're least worried. Okay, this person seems normal, this person seems normal. Here, these two people also seem to be very, very normal. They are just carriers. They don't show any symptom. You and me can also be a carrier. Now, when they have baby, there is a chance, 25% chance that the baby can have the disease, which is why we all should technically get ourselves genetically tested before having a baby. Now, obviously not for you right now, but later stage in your life, definitely yes. This is very, very, very important. Okay, this is important, just remember it. Okay, so this was it. Where is chromosome number? Okay, where is beta globin chain present? In which beta globin gene is present on which chromosome? Come on, just tell it, and then we'll move on to the next disease which is there. Swastik says, thanks ma'am. Thank you. Color of the eye and all, it's all genetics. Yes, everything, each and every part of our body is basically genetics. This, okay. Vidal says 11th chromosome. Perfect. 11th chromosome is where you have the beta chromosome. Perfect. Now I'm just reading it out for you. And by the way, this question is for you guys as a homework. People with sickle cell anemia won't have malaria. Is that a question? That question is for you guys. Okay, you have to check it out. Okay. Anyways, sickle cell anemia is caused by mutation in the beta globin gene. Glutamic acid is replaced by valine at the position 6 due to GAG to GUG substitution out there. Mutant hemoglobin molecule undergoes polymerization. Mutant hemoglobin, which is made by the bad beta globin chain, it is going to what happen? It is going to get polymerized. It's going to come together under low oxygen tension, causing the change the shape of the RBC. Okay, so RBC has a biconcave shape is what you say. Biconcave disc-like shape is what is there, but now it turns into a sickle-shaped structure. Okay, reducing the oxygen transport and causing blood flow obstructions out there. Please remember, it is biconcave in shape, biconcave, and then it transforms into sickle cell. HBS, homozygous condition when both the alleles are same, the person will be having disease. If at all HBS, heterozygous, biheterozygous, both the alleles are not same over here, this is going to be a carrier. And finally HBA, both the alleles are same, but they are going to be normal genes, so it will be normal. Cool. Cool. That's all. That's all about sickle cell anemia, which is your first blood disorder out there. Now, it's a correlated disorder. Until now, we have spoke about two blood disorders. One was sex-linked, hemophilia. This is going to be the other one, which is autosome-linked, and I hope you know that. Now, you have one more disorder, which is there, and I am going to talk about that directly. That is thalassemia. That is going to be what? Thalassemia. It also works on the same principle. A little difference is there. A little difference is going to be there. What exactly happens over here? Again, hemoglobin has been affected. Ayyo. Hemoglobin is there. Hemoglobin is made up of what? Alpha and beta chain. 2 alpha and 2 beta. That is how it works. Alpha globin genes which are there, they are present on, as Amalu said, it's present on chromosome number 16. Okay, beta globin genes are presented on what? Chromosome number 11, as you both said, right? Now over here, look, the beta globin chain is just having one gene for it, one pair of gene, here and here, that's all. Okay, what about the alpha one? Alpha basically has 4 of them. Suppose I'm just making it here, it will be the top. This is one pair of gene, this is another pair of gene. So we say that this is HBA1, which has 2 alleles, and there is HBA2, which have 2 alleles. Okay, each of them can help in the formation of alpha globin chain. Similarly over here we have HBB gene. Okay, we have HBB gene for beta globin chain. The thing that I'm coming to talk about is that, okay, is very, very, Amalu, I'm going out, sorry for the leaving, it's okay, have a very good day Amalu. Please take care of yourself. Okay, the thing that I'm saying over here is that this disorder, that is thalassemia, is going to be a quantitative, not qualitative, quantitative disorder. What do you mean by quantitative? Here the quantity of it is going to be hampered. So I am saying that basically one gene is affected. What is going to happen? Your beta globin chain is going to be produced. Your beta globin chain is going to be produced, but the amount is going to be less. Okay, what if both of them are gone? Basically, beta globin chain which is there, it is not going to be produced at all. If it is not being produced at all, do you think hemoglobin will be produced? No. Okay, similarly over here, if suppose like this scenario is there, alpha globin chain is definitely going to be produced in this scenario. Suppose if one is affected, what is going to happen? Still it is going to be produced, but the amount is going to be less. What if two of them has been affected? Still you have other two to do their work, which is why it will be still less. Suppose this is also being affected, then what happens? Then it will be very, very less. Suppose this is also affected, no alpha globin chain is going to be produced. So in this scenario, what are you noticing? You are not noticing about the other thing. You are not noticing about the quality of hemoglobin being formed. The quantity itself is not there. If alpha and beta globin chain is not being produced enough, do you think hemoglobin will be there? If hemoglobin is not there, do you think people can survive? No. Why? Because oxygen is required. So people can have thalassemia. There are thalassemia minor patients, there are thalassemia major patients out there. I'm going to talk about the differences that you have so that you get an idea. Suppose in alpha thalassemia, okay, in alpha thalassemia, if only one particular gene is being affected, I'll say that that person is going to be a carrier and that person will not show any symptoms. Why? That person should not show any symptoms. Why? Because over here you have three other genes out there which is going to show their effect, which is going to produce enough amount of alpha globin out there. On the other hand, if at all you see a scenario like this where both of these genes are affected, still you have other ones, I'll say this is going to be alpha thalassemia minor patient. They have mild thalassemia. They have very mild thalassemia. They might show a very less symptoms out there, but they will be having it. Okay. Now over here, a person with 3 mutated alleles. A person with 3 mutated alleles basically means only one is there to take over things. This is known as HBH disease. In this particular case, people might not survive until a particular age. Okay, until a particular age, for example, within 10 years of age, they might die. They might die. That is usually how it works. Now over here, this is a scenario where basically all the genes are being mutated. This is known as alpha thalassemia major case, and this usually happens, when this happens, usually in the birth, when a baby is being born, after a few months itself, if at all it is not being diagnosed, if at all blood transfusions are not being given. If you give blood transfusion, what is going to happen? Basically, you're getting hemoglobin from somewhere else. You can still survive. But people might not know about it, which is why the baby might die. The baby might survive for some time, reason being that, you know, when the baby is born, the baby has fetal hemoglobin. In the fetal hemoglobin, as in the fetus, baby is known as fetus, okay? Fetus have their own form of hemoglobin, which is known as fetal hemoglobin. They are transferred, or basically they are changed to the adult form of hemoglobin. So while that change happens, my dear children, they do not have anything being formed, which is why, as I said, they could die as and when they are born. So it varies depending on how many number of genes are being affected. It is definitely going to be varied. So, ma'am, what happens when a person inherits one mutated gene from a parent in a heterozygous state? In a heterozygous state, they might still survive, as I said, but they might be a minor patient. Okay. Swastik, ma'am, I am American born. Should I give KCET next year? Dear, it depends upon your parents. If at all your parents have studied in the state of Karnataka for 7 years and things like that, you can definitely go and give KCET examination. Okay, Viral says they are mostly immune from malaria, but they can be affected by malaria. Viral, yes. Yes, they can. Okay, you're talking about sickle cell anemia, right? Right. So listen to me carefully over here. So this is a scenario that we have in the case of thalassemia. And about thalassemia, they have not given too many things. Whatever is given, I have written over here in your NCERT. So they have mentioned that a mutation or deletion reduces synthesis of either alpha or globin chain of hemoglobin, leading to abnormal hemoglobin and anemia. Alpha thalassemia is where it affects the alpha chain production caused by mutation in the HbA1 or HbA2 gene on chromosome number 12. Beta thalassemia is where it affects your beta globin production caused by the mutation in HbB gene on the chromosome number 11. On the chromosome number 11. So that is what is there. And Amitabh sir is having the disease? No, he does not have the disease. He is just a carrier. A carrier. So being a carrier is different. Being a carrier means, suppose you and me can be a carrier. Okay, always understand. You and me, both of us can be a carrier. We might just transmit the disease to someone else. We might not have it. You get it? So Amitabh Bachchan sir is a thalassemia carrier. Always remember. So you don't have to remember that, but I'm just telling you that carrier options can be there. So for example, as I said, over here, this person is going to survive because they have other two genes doing the whole thing. So his son have it? No. Again, if his partner does not have the particular gene, then it's not there. But the son might have the gene. The son could be having the gene. Suppose over here, if Amitabh Bachchan sir is there and over here these two alleles are being affected, these two alleles are not. This chromosome went. Suppose this chromosome went in the gamete and from the mother's side also he didn't get anything. What happens? There is no chance that that other son which is there, he can be a carrier. He might not be a carrier. What if this went and from the mother's side a normal thing came? Then he is also a carrier. Then he is also going to be a carrier. You get it? No one can survive with thalassemia. What I mean is, if at all, even if you basically undergo repeated blood transfusion, depending on the severity of your case, yes, people can survive with a lot of blood transfusions out there, but only up to a certain limit. Most of them, they die. You get it? That is usually the case. So I'll tell you my own story. Okay, so I have lost two of my cousins because of thalassemia. Okay, and both of them, they survived. I think I have told in one shot also. Both of them, they survived only until the age of almost 10. One person was almost my age. So with that, I got an understanding. Okay, back then we didn't know that, but right now we know it. Okay, so over here, what exactly happens is, the father, that means my uncle, probably would have been a carrier. Their wife, okay, his wife who is there, she also might have been a carrier, which is why both their children had the disease, and basically both of them could not survive. So we all can be carriers. We all look very normal. We can, suppose I can be definitely a carrier. I have not done my genetic checkup, but I can definitely be carrier. And if at all you grow up, probably you speak to a lot of people out there. If they have done the genetic checkup, they might be knowing they can also be a carrier. So this is very, very important. We do have to check. Now I can be a carrier, I may not be. My mother definitely could be a carrier. My mother definitely could be a carrier. Okay, so that is something that is there. And yes, my brother have done the checkup and he is a carrier. So that means my mother is definitely a carrier out there. So right, so that is how it works. And yes, we all look very, very normal, right? But in our body can survive with a little amount of hemoglobin and things like that because it is produced amply. There's not an issue. But when scarcity of hemoglobin, that's when we say it's a major patient of thalassemia and that they cannot survive for a very long time. So yeah. So yes, sorry for your cousins, ma'am. It's okay. And Viral, how do you know that Amitabh sir is a carrier? Google. Okay, so I was previously, when I was making the PPT, I think the previous year, that's when I just went through, because I was very curious to get to see this. Okay, when you know, Queen Victoria, who is there, she was also a carrier for hemophilia. She did not had hemophilia. She was a carrier for hemophilia. I read that somewhere, and basically I was curious to find, is there someone else also? That brings attention. And you get to understand more about the disease. So just for making you aware about things. Yes, definitely. Before marriage planning, or basically where you plan for kids, definitely we should all do a checkup, a genetic checkup. Swastik, man, there are no good teachers in Florida like you. Oh my God. Thank you so much. It means a lot. Okay. So I hope that works. Okay. I read that Queen Elizabeth took her blood with her whenever she goes. I have no idea about that, June, but that's a good thing. Probably I will go and explore more about it. Okay, so I hope this particular thing is also clear to all my children out there, and I can come back to the topic. The second autosome linked disorder. So basically we have completed two autosome linked disorders, sickle cell anemia and thalassemia. Okay, thalassemia is a quantitative disorder. The quantity of hemoglobin is being hampered. Always remember, quantitative has a T and thalassemia also has a T. You can remember like that. Cool. Now over here, now we have what? Now we have the next disease, which is phenylketonuria. This is the third autosomal linked disorder. Basically here the second one, because in your NCERT that is how it goes. So can we go ahead? Okay, can we go ahead and talk about it? If it works, just give me a thumbs up. And yes, your curiosity makes me so happy. You know what, I would love to see you guys in all the live classes if at all possible. Seriously, I mean, it's so good. Okay, I'm gonna research about it. Okay, so phenylketonuria over here, what exactly happens over here? Again, an autosome is being affected, a gene on the autosome is being affected. What exactly takes place over here? Suppose over here I have a gene, say I have a chromosome, I have a gene, and this gene codes for an enzyme that is known as phenylalanine hydroxylase. Phenylalanine hydroxylase. This is the enzyme that it codes for. What exactly happens over here is that there is some mutation happening in this particular gene. There is some issues happening in this particular gene, which is why the enzyme is not being made. It's not being made. Now, what's the issue with that, ma'am? What's the issue if at all this particular enzyme is not being made? The problem that is going to be caused over here, listen to me very carefully, is that in our body, you know, whenever proteins, they are broken down, we get amino acids, because amino acids makes the proteins. So when proteins are broken down, we get amino acids, right? And these amino acids are being used for several functions. So we have one amino acid which is known as phenylalanine. Okay, phenylalanine. When this enzyme is there, phenylalanine is able to convert itself into another type of amino acid which is known as tyrosine. Okay, now suppose this enzyme is not there, this conversion is not happening. If this conversion is not going to happen, what is the problem with that? It stays in our body either in the form of phenylalanine, or probably it stays in the body in the form of phenyl pyruvic acid. Is that a problem, ma'am? Yes, because if at all it stays in our body in a high amount, basically it can lead to mental retardation. It can lead to what? Mental retardation, which is why it is going to be a very big problem. Okay, ma'am, in this case, the urine deposits have high level of phenyl pyruvic acid, right? Exactly. So, you know what? Kidney cannot reabsorb this. Usually, kidney reabsorbs a lot of things which are there, which are moving out of our body. It says that, oh my God, I need water, let me take about water. Oh my God, I need glucose, let me take about glucose. A lot of things happens. It is reabsorbed. But phenyl pyruvic acid, it says that I do not want that. Please take it up. Okay, so please let it go from the body. So what happens is that even though it's accumulated, just understand that its accumulation is also more. Okay, even after that our body does not wants it also, basically it moves, let it go out, and as it passes out through our urine has a very foul smell for the same reason, because phenyl pyruvic acid is basically moving out. Okay, so that is one problem, that is mental retardation. And as I said, along with that, since our body does not absorb, or basically our kidneys does not reabsorb this phenyl pyruvic acid properly, which is exactly why they move out of our body, and that's why we have a foul smell. Cool. That is one character being affected. If you remember, this particular disease was an example of pleiotropy. If you have attended my live classes before, you might be knowing about it. Pleiotropy is basically where, generally what did Mendel said? One gene, one particular character. One gene is basically responsible for one particular character. But in this particular case, one gene might have multiple effects. This is one effect. There can be multiple other characters that is controlled by this particular gene, and we're going to talk about that right now. So when this does not happen, obviously you have a problem being created, but there's one more problem. Tyrosine is when it is not being made. No, there is another pigment in our body which is not being made. It is known as melanin. It is known as melanin, and melanin is responsible for your hair pigmentation and your skin pigmentation. If melanin is not being produced, what will happen? You will have reduced hair and skin pigmentation. You will have reduced hair and skin pigmentation happening up there. So two characters are being controlled by one particular gene itself, which is why it is an example of pleiotropy also. Cool. Now June is asking me something. So ma'am, if the people didn't pass urine and they have renal calculi, type of stone in their kidney, ma'am, will they die? Oh my God. Oh, I do not know, but the type of the stone, will they die? They might not die. It depends, you know, whenever someone has stone in their kidneys, obviously the urination thing is going to be very painful, and you go diagnose it very early. You start drinking water, and people generally do not die of a kidney stone. Okay, sometimes appendix and gallbladder stone and stuff like that can cause issues, but kidney stone can be treated very, very smoothly. So it's something that is not to be worried about, but you have to be careful. Cool. So this was about phenylketonuria, which was again an autosomal linked disorder, which is there. And what is said over here? Lack of phenylalanine hydroxylase enzyme. It prevents the conversion of phenylalanine to tyrosine. Effects is what? Accumulation of phenylalanine and phenylpyruvic acid leads to mental retardation. Apart from that, these compounds are excreted in the urine due to poor kidney reabsorption, which is why it gives a foul smell as well. No, ma'am, I'm asking that people with phenylketonuria having renal calculi. I have to check that. I don't think. Okay, I don't think that could lead to that, because as I said, they definitely will show symptoms. But yeah, would check the severe cases. Okay, so yes. Is this clear? Is the third one also clear to all of you? Perfect. Viral? Okay. So now here is a question that I have from your KCET point of view. Yes, here's the question. Identify the odd one among the following disorders. The first one is hemophilia, the second one is sickle cell anemia, third one is phenylketonuria, and the D one is thalassemia. You have to tell me which is going to be the odd one out. Which is going to be the odd one out? Can you tell me? A, B, C, D. Hemophilia, sickle cell anemia, phenylketonuria, or thalassemia. Listen over here. Hemophilia is a sex-linked disorder. Right? June says A. Yes, A is the answer. Sickle cell anemia, thalassemia, and phenylketonuria, three of them, they are going to be autosomal disorders, which is why the odd one out will be this one. But some people can think it otherwise also. They think like this. Sickle cell anemia, hemophilia, and thalassemia, they are blood disorders. In that case, phenylketonuria will be out of the thing. Okay, but just understand that there are a lot more similarities when you talk about these three. As I said, they are autosomal disorders, which is why A remains to be the answer. Okay, A remains to be the answer. This had actually come before. Now, one more question is there. Example for non-Mendelian disorder. Example for non-Mendelian disorder. Thalassemia, hemophilia, cystic fibrosis, or Down syndrome. What do you think is the answer for this? Come on, try this also. Example for non-Mendelian disorder. Mendelian disorder, you know. So you have been asked which one is not a Mendelian disorder. That's a simple question. Okay, thalassemia, hemophilia, cystic fibrosis, or Down syndrome. Rekha is here. Hello, Rekha. Rekha came in with a banger. Says D is the answer for this question. Down syndrome. Is it D? Is it D? This is KCET 2022 question. Is it D? Yes, it is going to be D. Why? Because this, and this, all of them are Mendelian disorders. But ma'am, cystic fibrosis we didn't study. Listen. So basically, Down syndrome is definitely cystic fibrosis. Actually, comes under autosomal recessive disorder itself. Autosomal recessive disorder. In your NCERT, there is one line given that it is autosomal recessive disorder. There is no explanation about this, but please don't know. Okay, so D is going to be the answer. Yes, June. Yes, Rekha. Perfect. Down syndrome is a chromosomal disorder. We're gonna soon study about that. Now, over here, I have a question. With whatever we studied until now, we probably can solve this question. This was a previous year model question paper question. In this year also, they have repeated some same model question paper, right? So over here, probably we can try this and try to find the answer. One for each of the answers. Okay, one by one, we're gonna do. Sex-linked recessive disorder occurring in about 8% of males and only 0.4% of females, leading to defect in the cone of the eye. Which is that particular disease? Which is that particular disease which affects 8% of males and 0.4% of females, and it affects the cones of the eye, cone cells of the eye. Come on. Which is that Mendelian disorder that we have? Waiting. A is color blindness. Perfect. A is going to be what? Color blindness, because how you can identify it? Again, the males are being affected and females are less affected, and not just that, here you're talking about defect in the cone of the eye. So it has to be color blindness. June also said color blindness. Perfect, Rekha and June. What about B? Sex-linked recessive disorder affecting a single protein involved in the clotting of blood. Involved in the clotting of blood. June says B is hemophilia. B is hemophilia. Yes, hemophilia. Why? Because hemophilia is a disorder, a blood disorder, where the clotting is being affected. Right? Yes, Rekha also says B is hemophilia. Perfect. Macha, everything is clear. Then I am good. Okay. Inborn error of metabolism, autosomal recessive trait, accumulation of phenylalanine. Accumulation of phenylalanine. C is phenylketonuria. Perfect, June. C is going to be what? Phenylketonuria. Why phenylketonuria? Over here you see phenylalanine is being accumulated, and not just that, it's an inborn error of metabolism. That means it's there from the starting point onwards. From the starting point onwards. Right? Yes. Rekha says also phenylketonuria. Perfect, dear. Next disease. Let's see. Rekha says thalassemia. June says sickle cell anemia. Let me check. Okay. Autosome linked recessive blood disorder where substitution of amino acid in the beta globin chain of hemoglobin occurs. Substitution. Substitution means basically it was GAG. Now it is substituted to GUG. Right over here, glutamic acid was there. Now it is valine. Baba, this is the case of sickle cell anemia. This is the case of sickle cell anemia. Okay, and the last one which is going to be there, autosome linked recessive blood disease, reduced rate of synthesis of globin chains of hemoglobin. Reduced rate of synthesis. Reduced rate as in you're talking about quantity, right? So it has to be quantitative disorder, which is going to be thalassemia. But I'm really happy. I think all of you most got all of them correct. Okay, one or two here and there was wrong, and that's totally fine. I am super proud of you. Perfect. Perfect, perfect. Okay, so Rekha and June, you guys aced it, my dear children. I'm so super happy for you. Okay, now talking about the next question which is there over here, this is again a model question paper question. This is 2024-25 question. So they had mentioned like this: few autosome linked recessive gene blood diseases occur in human population. Among them, some are related to qualitative and some are related to quantitative problem of blood proteins. So how do you identify that and write it? Basically you mention qualitative disorders are where the quality of the hemoglobin is being affected. Okay, quantitative disorders, blood disorders are where the quantity of hemoglobin is being affected, and you talk about that like that. Okay, so you mention these problems can be categorized as qualitative, affecting the structure or the function of the protein, or quantitative, affecting the amount of the protein which is being produced. Sickle cell anemia is going to be a qualitative problem. What is going to happen? Mutation in the HBB gene on chromosome number 11 causes the production of abnormal hemoglobin, which is maintained by HbS. Okay, this defective hemoglobin leads to sickle-shaped red blood cells, impairing oxygen transport and causing blockage. This is something that we have studied. So you just have to write it in points. Only if you write in points, you will get marks. Similarly, thalassemia is a quantitative problem caused by mutations in gene responsible for hemoglobin synthesis. It can be HbA1, HbA2 in alpha chain and HbB for the beta genes. There can be mutation in one allele or multiple alleles are there. Depending on that, it can be a quantitative disorder. Can you take a screenshot? Yes, definitely. Dear, you will be getting the, you can get the PDF of this in our WhatsApp channel as well. You just have to join the WhatsApp channel. If you want to move out of it anytime, you can do so. Also, I spoke about the page, right? AIO page. There also the PDFs are being uploaded. So you can get the whole PDF of the entire thing if you wish. And you can take the screenshot, by the way. I'll just move aside. Okay. So I hope that works, and now I go ahead. So explain thalassemia as an example of Mendelian disorder. This is MQP2 previous year question. Okay, previous year question over here, and you have so many things. June, are you able to find the PDF up there? I hope yes. If not, probably I have to report. Yes. And join the WhatsApp channel also. There probably we can talk more. So thalassemia is up there. You just have to talk about everything about thalassemia that you know right over here. Again, if you wish to take a screenshot, you can. The disorder occurs due to mutation or deletion of genes involved in hemoglobin synthesis, leading to reduced rate of synthesis of alpha or the beta chains. The beta globin genes are located on this chromosome, and mutation or deletion of one or more of these can reduce alpha globin production. Similarly, this is the beta globin gene. It is there on chromosome number 11. If at all there is mutation in one or both, it affects beta globin synthesis. That's all. Simple things. Cool. Yes. Now we come to the next topic, that is pedigree chart, and this basically makes everything a big headache for a lot of children. So what exactly is pedigree analysis? Pedigree analysis is basically we are trying to understand how is the flow of trait going throughout each generation. Throughout each generation, we are just studying about how is exactly everything working. This is known as pedigree analysis. Okay, so ma'am, dear, I am making you solve KCET pre-YQs along with the concept. So for example, when we do the pedigree done, we will be coming ahead with KCET PYQs. So the red ones which you see over here are all KCET PYQs are there. Okay, so after each concept, we are solving a question. Works? Okay, now the question that you solved was also PYQ question, by the way. Okay, now listen to me. In pedigree chart, what is the thing that you need to remember? And it goes like this. First is, this means a man is there, a normal man. This means a normal female is there. This means affected man is there. This means affected female is there. Okay, suppose you have something like this, that means sex is not being specified, whether it's a male or a female. Suppose it's mentioned like this, that means 5 children are being born. We do not know whether it's a male or a female. Okay, suppose it is like this, that means 5 of them are being affected. If at all you see something like this, it means that this is mating between a male and a female and a male. And if at all you see something like this is very important. This is the most important one. If you see something like this, it means it's a mating between two relatives. Okay, 2 relatives. This is very important. It's known as consanguineous mating. Okay, you also call this as consanguineous mating. How do you remember it? 2 lines are there in between. That means too much amount of closeness is there. You can remember like that. Okay, so 2 lines are there. This means consanguineous mating or mating between the relatives out there. Very important. If at all you see something like this, that means father and mother is there, and you have a son who is being affected. Okay, this is a basic thing that is there for all. And now we're probably going to see certain things as that things become all more clear. So until now, okay, is it highly dangerous for passing genetic disorders, ma'am? Yes, definitely. It's very dangerous. You can give KCET if at all you want engineering colleges in the state of Karnataka, or probably you can look for the IITs also if at all you wish for that. Swastik. Okay, so yes, let's come back. So over here, listen to me carefully. Okay, these are 2 pedigree charts given to you. One is of sickle cell anemia, which is an autosomal recessive disorder. In your NCERT, you could not spot a dominant disorder. Did you until now? No. Sex-linked recessive disorder, color blindness, hemophilia, autosomal recessive disorder, sickle cell anemia, thalassemia, phenylketonuria, and then cystic fibrosis, about which I spoke. There is no autosomal dominant disorder. Wrong. In your NCERT, under the pedigree chart, that is this, you have been given an autosomal dominant disorder, and the only autosomal dominant disorder which is going to be what? Myotonic dystrophy. Which is going to be what? Myotonic dystrophy. This is very important. Now, how can you understand whether it's autosomal dominant or basically autosomal recessive and things like that if you're given a pedigree chart? Because you get pedigree chart related questions. So listen to me carefully. Over here, if at all you see a father and a mother is there, right? This is the next generation, this is the next generation. In every single generation, if one person is being affected, you will say this is a dominant disorder. What do you call this? This is a dominant disorder. Why? Because every single generation, one person is being affected. Here, the mother is, here, again the mother is, here, the daughter is. Right? In every single generation, it's there. On the other hand, if at all you see something like this, here the parents seem not to be affected, but the daughter and the son are being affected. You see something that is known as skipping of generation. What do you see? You see something that is known as skipping of generation. It is being skipped. Here also the parents are not being affected, but here you see it. Why? Because it's a recessive disorder. So first rule of pedigree is understand, if it is not skipping, Baba, it has to be a dominant disorder. What is it? It is definitely going to be a dominant disorder. If at all you see skipping of generation, it is going to be a recessive disorder. And this is not something for mugging up. Listen to me carefully. If you see, Baba, if over here I say this person, okay, I should not be, okay. If I spoke about sickle cell anemia, it was HbA and HbS. HbA, HbS. Both of them were normal. They had a defective gene, but it was still fine. Why? Because the other gene was overshadowing the effect. The parents were normal. But when this and this came together, you saw that basically the son or the daughter could have the disease, right? That is what I mean. Here, the parents could be normal because the other gene is there to overshadow the effect. Uttam, DA, okay, you're talking about dominant autosomal, sorry, I'm not getting. Okay, so that is the logic. Now, how do we understand whether it's autosomal or basically sex-linked? In your NCERT, this autosomal disease is given. How do you understand whether it's sex-linked disorder? So listen to me. In sex-linked disorders, generally we see males are being affected. Here, if at all you see male and female equally being affected, right? So if it is sex-linked, mostly males are being affected. Not just that, you're going to see a crisscross pattern. What do you mean by crisscross pattern? You will generally see something like this. Suppose a female is being affected. In that particular case, her son is going to be affected. Then this son, when he marries, the daughter is going to be affected. That is what you mean by crisscross pattern. Female, male, female. Similarly, male, female, next male. That is what you mean by crisscross pattern. That is what you see. Okay, that is what you see in the case of sex-linked disorder. And sex-linked is usually not being asked, but you should know it. What if they ask you? Okay, generally this has been asked. So here, if at all you see, here you do not know whether it's autosomal or sex-linked. If you look, male and female, both of them equally affected. From female, female is getting affected, so this is not crisscross that I am seeing over here. Similarly over here, male and female is being affected. Here basically none of them are. Here you do not know. Here you do not see female crisscross pattern or things like that. Again, which is why I'll say it could be autosomal. Got it? Now, when I make you solve a question, probably we might be able to do better understanding. We might be having a better understanding of this. Here is a question. The affected male in the pedigree chart is symbolized by. This is a simple question. We will go to the advanced version of this also. The affected male in the pedigree chart is symbolized by. You have this, 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 and this. A. June says A is the answer. A is the answer. Is A the answer? Affected male in the pedigree chart is symbolized by a square which is being circled. But ma'am, in females, the affected one, why not showing the symptoms, whereas the male shows? I didn't get it. If the female is affected, she will be showing the symptom. If the female is affected, she will be showing the symptom. If at all it is X-linked disorder, there is another X chromosome to overshadow the effect. In that case, she will not. Okay? Okay. Now over here, I have a question. Again, in the following symbols used in human pedigree analysis, identify the symbols that denotes consanguineous mating. Consanguineous mating, as in mating between what? What do you say? Family members. Mating between relatives. What do you think is the answer for this? Uttam, come on, try for this. June, also you can. Go ahead, go ahead, go ahead. Okay, B. B is what you say. B is what? B is what? B is going to be this one. Yes, it is going to be B. Why? Because you have two lines in between. Answer is B. Perfect. Cool. Done. Now try to solve this one. This is where I'll need the attention. From the following pedigree chart of the family 1, which looks like this, what does it show? This is a trait that we have. What does it show? That is a question. Over here, it is an autosomal dominant trait, it's an autosomal recessive trait, it is an allosomal dominant trait, it's an allosomal recessive trait. Allosomal means sex-linked. Allosomal means sex-linked. Now, probably we can try to find the answer. Waiting for the answers. Others can also try, okay? I'm just waiting for you guys. B. Okay, it's coming very late. Okay, so B means this. Okay, let me check it out. It is an autosomal recessive trait. Is it? Let's check it out. Recessive or not? First, we have to check that. In this generation, it's not there. In the next generation, it's there. So the skipping of generation is being seen. So it is definitely recessive. Now, it is autosomal or sex-linked. That is the one that we have to find out. Males and females equally affected. Here, females are being affected more. And there is no crisscross pattern, which is why autosomal. And yes, June, you are absolutely correct with that. So the answer for this question is going to be what? B. It's an autosomal recessive trait. Perfect. And I think I have a recent question as well. No. Okay, no, this is 2016 question. Now here, identify symbols used in pedigree analysis. This was previous year model question paper based question. What do you mean by this? Consanguineous mating. What do you mean by this? Male and female mated, but they have an affected child who is a son. Affected male. This is a very simple answer. This was a 2-marker question that had come from the chapter Principles of Inheritance and Variation. Okay, I don't think there's too much explanation needed for that, because there is one question which is actually very important, and I want all of you guys to basically try for that if possible. Come on. Here the question goes like this. Okay, the question is actually says that pedigree symbols are used to analyze the disorders in human population. Okay, we have to mention the genotypes of the following pedigree symbols. If at all I am saying it's an autosomal dominant disorder, I am saying it's an autosomal dominant disorder, and basically I am making a square which is being affected. That means the person has the disease. It's an autosomal dominant disorder and the person has a disease. How will you show the genotype? If I give you a clue that basically this is the dominant allele and this is going to be a recessive allele. With the help of it, can you tell me what will the genotype, genetic makeup of this particular organism? Any guesses? Any guesses which is there? It's actually very important. I'll solve one question and probably you might be knowing it then. Okay, when I say autosomal dominant disorder, that means that, and if a person has the disease, that means basically they should at least carry one allele that is going to be dominant. If they have something like this, basically it will overshadow the effect of this anyway and the person will have the disease, right? Or the person can be like this also. Even having one dominant allele is fine, or the person can have something like this. Anyway, the person will be affected. Got it? Capital A, small a, or capital A, capital A. That is a general scenario that we have. Let's talk about autosomal recessive. Is this clear? Is the first one clear? June, I see the answer out there. Good. So capital A, or capital A, small a can work. Now we are going ahead with the second one. Can I? If yes, can you give me a thumbs up? If yes, if it works, can you give me a thumbs up? Yes, listen. So autosomal recessive disorder over here, this is a female who is not being affected. Okay, if she was affected, how will you show it? First think about that. If she was affected and it's a recessive disorder, you will show like this. You are going to show like this. We are not talking about 6 chromosomes over here. Why? Because it is an autosomal recessive disorder, right? So I'll say 2 alleles are there, both of them are same, and both of them are recessive. Then only the person will be having the disease. Otherwise, she will be a carrier and things like that, right? So this person is having the disease. In that case, it would have been colored. But here, that's not the case. The person does not have the disease. So how will you show it? We have to make sure that the recessive alleles does not come like that together. It can be either again like this or like this. Why? Because this will overshadow the effect, and here you do not have the small allele at all. You get it? Again, capital A or basically capital A, small a. Perfect. Now we have sex-linked dominant disorder, and you have something like this which is not being colored. Sex-linked dominant disorder, and this is your dominant allele. So XA is dominant and basically XA is going to be recessive, if at all you want to think like that. How will you show this? Sex-linked dominant disorder. Sex-linked dominant disorder. And this is a male. So male, for that male, I'll put XY. And over here, I do not have to put anything. That itself is fine. That itself is fine. Or if at all I want to put, I'll put the small allele, small a. I'll put the small allele. Exactly, June. Perfect. Works really good. Super proud of you. Last one, sex-linked recessive disorder over here, and basically both of them are equally affected. Male and female are being affected. How will you show that? And it's a sex-linked recessive disorder. So you make like this, you make like this, and you're saying both of them are affected. That means one has to have the small allele here in the case of man, and here in the case of female, both of them would be having the allele. Got it? Which is how you get your answer for this also. It's a little twisted one, but yes. XAY and XBY. Didn't get it. XAY and XBY. This is a female, right? Got it? For male, okay, for male X. So why are we using actually B term? Why are we going ahead with B, small b? Because until now we were talking about capital A and small a, right? So we just can go with the small a one. That works. Or that could be creating confusion with your teacher. So this is how it basically goes. Cool. Works. Let's go. So this is how it is, and these are the type of questions that was there. And finally, we have chromosomal disorders as well. Ayyo, it's raining. Last topic is there, chromosomal disorders, and shall I go ahead and teach you? I'll be just here for 10 more minutes, so I'll try my best to do as much as I can. Probably we'll be doing next thing in the next class, if possible, okay? So listen to me very carefully. We have chromosomal disorders, and I told you chromosomal disorders are being caused. Suppose when I say that over here, it can be caused because if I have a chromosome, a pair of chromosome, now I have one more extra one that causes a problem, I'll say it's a disorder. Suppose I should be having two, at least two of them, but now I just have one. That is leading to a disorder out there, right? So all these type of things contribute to disorder, which you call as chromosomal disorder. There are two reasons for it, okay? One is aneuploidy and one is polyploidy. Aneuploidy basically means failure of segregation of chromatids during cell division cycle, leading to the gain or loss of a chromosome. Now here, the most important thing that you need to remember is that basically it's caused due to either you are getting an extra chromosome or you're losing a chromosome. That is what you mean by aneuploidy. Why it is happening? Probably because the chromosome which was there, so you have a pair of chromosome over here, and when they make a gamete, they have to segregate, and one goes to each of the gamete, right? That is how it usually works. Suppose here failure happens in the segregation, in the separation process, both of them will go to one particular gamete. Okay, another guy will be having one gamete, the male carries one gamete. Over here, the female carries a gamete that has two chromosomes. What happens then? Then you get 3 chromosomes. That is a big problem. Okay, this scenario can happen, or in this scenario, in the other chromosome, in the other gamete, Baba, since both the chromosomes went over here, you do not have anything to put over here. When this chromosome, when this particular gamete fizzes with that one, what do you get? You get only one chromosome. The other one is actually missing. So that is what you mean by failure of segregation of chromatids during cell division. That usually happens in us. Now the next scenario is polyploidy. Polyploidy is actually seen in the case of plants and all. Okay, what happens there is that suppose over here a cell is there. A cell has n number of chromosomes over here and here. This cell was ready to be divided into 2. This cell was ready to be divided into 2. That is why it was doing all of it. Okay, it was duplicating the number of chromosomes and all. It increased the number of chromosomes in a way that is going to divide in 2. But now this division did not take place. If at all, Baba, it is not going to divide. Okay, if at all it is not going to divide over here, what is going to happen? If it is not dividing over here, the next thing that is going to happen is that you get a whole set of chromosome as an extra one. You get a whole set of chromosome which is extra over here. So failure of cytokinesis. Cytokinesis means cell division. Failure of cell division after telophase stage. The telophase stage is the last stage that we have in cell division, results in an increase in the whole set of chromosome, and that happens usually in the case of plants, not in our case. Usually in the case of plants and not in our case. Got it? Works? Just give me a second, okay? Just give me a second. Okay. There is some noise that came in the, what would you say, in the AC. Guys, I think it's raining outside and I should be stopping this particular live. Okay, I think there's some issue with the studio out there. Okay, now the thunder is basically some electric wiring issues are there. I feel there's some noise and all coming. It will be safe if at all I move out right now. So will it be okay if at all I come again in the next live and probably talk about the chromosomal disorders? Oh, by the way, there's one more thing. You do not have to wait until the next live. So listen, over here, so basically, if you go and check out a playlist, okay, I have done the one-shot videos for almost 5 chapters now. Okay, there is molecular base of inheritance, there is principles of inheritance, there is reproductive health, there is human reproduction and sexual reproduction in flowering plants. All of it, one shot is there. Make sure you go check. Okay? Works. So make sure you do that, and in that I have explained about chromosomal disorders. Or if you can wait, my next live is going to be there on next Wednesday. Cool, because we have live classes in the paid batches, that is exactly why. But ma'am, no class on the app? No class today, dear. Tomorrow there will be my class. Cool. So thank you so much, children, and I'm so sorry I have to leave you like that, but I think it's safe for me to go at this time, or something could be happening here. Okay, so Wednesday I'm waiting. See you soon, but watch the one shot, it could definitely help you out. And bye bye, lots of love, thank you so much, and please take care of yourselves, and had lots of fun. June, thank you for being a part of this class, and everyone else, thank you. Bye bye. Ma'am, any suggestion for bachelor students for Pariksha Veera? Every single day, target the classes and complete it. Or basically, you might be seeing topic-wise data also. If you feel that you are not good in that particular topic, go and watch that particular thing, if it's possible. Okay, instead of watching the entire thing out there. Yeah. So yes, guys, I'll come back. Not situation is not good over here. So Rudra, hello dear, and bye bye. Thank you. Thanks a lot. Bye bye.